Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
Go above and beyond.
they become very good at what they do, but only
0.1%.
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
With Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. I have Domenico Otranto. He's a professor of parasitic diseases. He's also the head of the Department of Veterinary Medicine at the University of Bari in Italy. And we're going to talk about his research. So, Domenico, thanks for coming.
Hi. Yeah. Good morning. Buongiorno. And thank you for inviting me. It's a pleasure to be here today at the Finding Genius Podcast. Yep.
So tell me about your background. What got you into working with veterinary medicine and parasites?
So I'm professor of parasitic disease of animals at the Department of Veterinary Medicine at University of Bari. Bari is in southern Italy. But I also teach parasitology at the Faculty of Human Medicine. And I mainly work with my research team on parasites of pets of zoonotic concern, parasites which are transmitted by animals to humans and vice versa. And we also have a PhD course, an international one, in animal health and zoonosis, which is really important in this subject. And within the parasites transmitted to humans from dogs and cats, we are mainly focused on vector-borne diseases, which is quite a concern in a Mediterranean area. So I graduated as a veterinarian at University of Bari and obtained a PhD in parasitology some years ago, I would say. And I traveled also for research in France and in Germany, working on arthropod vectors and doing something or more that could have been done here in Italy. So this is my background.
So when you say vectors, does that mean mosquitoes, bites from other animals? Like, what are the common vectors for the parasites you see?
Yeah, vectors, it means all arthropods, which may transmit pathogens, viral, bacterial, protozoa, helminths, to any vertebrate host. And I mean mosquitoes, but also sandflies, which transmit Leishmaniasis, or also ticks, which are amongst the most important vectors because they transmit many pathogens of veterinary medicine and medical concern, but also lice, for example. So there are many vectors which may feeding on the host also transmit pathogens.
So there's a lot of parasites out there. Which ones do you focus on?
I'm focused mainly on Leishmaniasis by Leishmania infantum, which is a protozoan disease transmitted by sandflies, which are tiny insects, and also on filariates like Dirophylarimitis and Dirophylar repens, which are transmitted by mosquitoes. But I also worked on vector-borne diseases transmitted by tick-borne diseases. So I would say that the most important parasites here in the Mediterranean areas are Leishmania infantum, transmitted by sandflies, and also Dirophylaria. So these are the main topics of my of the research we carry out here in Bari. Yeah.
So we could start with Leishmania. You said it's transmitted by sandflies. What animals get it and what happens to them?
Yes. The main host for Leishmania infantum are dogs, but the Leishmania may also infect cats and other hosts like foxes and also rats, rodents, and they may also transmit. They may also infect humans. So these are the main hosts. Even if dogs are the main host and the reservoir of infection as well. We, for example, very recently found and reported for the first time the infection also in tigers in southern Italy. It was published in Emerging Infectious Disease. Was quite interesting because that demonstrated that where there is endemicity of this infection, all animal species, carnivores, may be infected by Leishmania infantum. So it's quite important.
Do people ever get Leishmania? And if so, how?
Humans may be infected by Leishmania, by different species of Leishmania. We have 22 species of Leishmania. Most of them are also zoonotic. I would say that the most important species is Leishmania infantum. So this is quite important to define and to clearly state that humans do not get infected by dogs, but they are infected through the sandfly bites. After that, these tiny insects had a blood meal on dogs. So pick the Leishmania on dogs, and after a biological life cycle which completes in the sandflies, they may eventually transmit the pathogen to humans. So this is quite important because we do know that preventing the infection in dogs may also be an important means for controlling the infection in humans. And this is quite important because just consider that in some areas of the world, there is the misconception of dog culling for controlling human Leishmaniasis, which is not true, is not the best means for reducing the risk of human Leishmaniasis.
So what happens when a dog gets Leishmania? What happens to them?
So that's an important question, because in endemic areas, we do not have clinical signs which are typical pathognomonic for this infection. We may have a range of clinical signs from asymptomatic animals which do not show any clinical signs apparently, to severely infected and severely diseased and animals. So, for example, in animals which display clinical signs, we may have alopecia, we may have weight loss, lymphadenomegaly, and an animal, a dog, is like and looks like an old dog. Even if it's, for example, 2 years of age, he looks like it is an old dog. This is typical in canine Leishmaniasis. But meanwhile, we may have many asymptomatic animals. So this is really an important concern considering the prevention of human Leishmaniasis, because even if in asymptomatic animals, even if for asymptomatic animals, the sandflies may have a blood feeding on these animals and then transmit the infection to humans. So it's really important to diagnose this infection in endemic areas and to know how to manage the control and the prevention of the infection.
Is the parasite multicellular? Is it single cellular? What is it?
Protozoa parasite. It is a unicellular parasite which usually replicates in the macrophages of hosts and as a mastigotes. And then, as soon as they are in the vector, they replicate by binary scission in more parasites with flagellum, which is promastigotes. The promastigotes are in the infective form of the parasite. Yeah.
So it stays in the macrophages of the dog. Does it go into other tissues, or does it stop the dog's immunity or ruin its immune system? What does it do?
Yes, and this is the differences in the presentation of clinical signs is due to the different immune reaction that any dog may exert to this to the infection. Indeed, these parasites causes an immune depression of the immune system in some way. And also we have also in hypergammaglobulinemia, which is an hyperproduction of antibodies. And so we may have different clinical signs related with immune complexes which deposit in different organs. So you see that these parasites may cause different clinical presentation, because it depends on the immune reactiveness of the host. And it's the same for humans.
Well, what is the treatment for dogs? Like you said, they seem like old dogs. I guess they have low energy. And do they look different? And how do you treat them? What can you do?
In animals, for prevention of the sandfly, for preventing sandfly bites, usually may have very good devices which are represented by pyrethroids in different formulations, colors, spot-ons, and these pyrethroids repel the sandflies and prevent the bites of dogs. So this is the best way for preventing the infection. Otherwise, we have anti-protozoal treatment in diseased animals, but I would say that in endemic area, it's much better preventing than treating animals, treating dogs. Yeah.
Do the dogs die of Leishmania, or just, what happens to them?
Yeah, sometimes if they are not treated, they may die. Yeah, it's quite severe infection.
So what are you hoping to figure out about this parasite and others? Like what aspect are you studying and why?
So for these parasites, we mainly work on prevention. Indeed, we had the opportunity over the last 20 years, to work with different preventative measures. And we had experience with the powerful of different preventative measures for reducing the risk of infection of Leishmania phantom in dogs, mainly in sheltered dogs, which is quite important. If you consider that we have a global population of domestic dogs of 703 million of animals, you may figure out how is important the prevention in endemic areas for these protozoa disease. And these animals, that's quite important to be considered, are not only infected or potential blood source for sandflies, but also for ticks, which transmit tick-borne diseases. And amongst other, we have bacteria, which are quite important also for human, like Rickettsia conori, the causative agent of the Mediterranean spotted fever, or Rickettsia rickettsii in North America, the causative agent of Rocky Mountain spotted fever. So it's quite important, the prevention is quite important in these endemic areas. And I will say that if you consider that the shelter dogs have the highest parasitic infection rate compared, for example, with shepherd or household dogs, is quite important also for in different socio-economic economical context to prevent this infection, because these diseases, Leishmaniasis, is the typical disease of poor countries or the poorest countries. So it's quite important, and they're included in the so-called neglected tropical diseases. So in some way, our work is also inspired by a social commitment in doing what we do. I mean, we are parasitologists, we are veterinarians, but we are much more, because when veterinarians and veterinary parasitologists do not just do coprology. Absolutely, I almost do not do coprology, but they work on different aspects of parasitic disease. And most importantly, our focus is on those parasitic diseases which are also transmitted to humans. So the social commitment is also quite important in our research as background of our researchers.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
So what are some of the novel things that are coming in your field? Are you able to do, deep sequencing of the protozoa, for instance, or what do you think is going to make a difference?
So we work usually on phylogeny of many parasites, vector-borne, those that we mainly study, and on geographical phylogeny, and also we have worked on some models for predicting some specific parasites. I think, if you like, I can give you an example.
Yes, go ahead. Yes.
One of the main parasites that I studied over the last 15 years is Telatia callipeda, which is an eye worm that was known until 1989 as Oriental eye worm. Is a worm which localizes and perpetuates in the eyes of dogs, cats, and also of humans. So this parasite was known as eye worms, Oriental eye worms, because it was found in Southeastern, was prevalent in Southeastern Asia for a long time. And we found that the first spot in Europe, in southern Italy, of these Telatia callipeda eye worms in dogs, in some rural areas in southern Italy, with prevalence of infection up to 54% in some villages. So our first question was whether it was just a coincidence in that area, that remote area, or was a parasitic disease endemic for Europe, for the whole continent. And we immediately faced with a problem because we didn't know which was the vector, the insect which vector is nematode. And so we started our researches, and within 5 years in the field, we discovered that this vector is represented by a teeny, very small Drosophilid, which names is Fortica variegata, which is a Drosophilid which feed around the lacrimal secretions of dogs, cats, and also humans, and transmits the infective stages, the third stage, the larvae of these parasites. That time, when I started working on this parasite and I found the vector of Telatia callipeda, the infection was present and was reported only in southern Italy. But with some mathematical models, we predicted that many areas of Europe would have been infected by these parasites because they were suitable for the development of this Fortica variegata. So, after about 20 years today, we know that we have Fortica variegata in Spain, France, in Portugal, in Germany, in Eastern countries in Europe. So today we know that that Telatia callipeda infection is also a European reality. Telatia is an European worm, with many cases also in humans. So this is an example on how the research and our research is impacted on knowledge of this parasite, and most importantly, we predicted that would have happened in the future. Only because the biology is very simple, and the vector Fortica variegata is present all over Europe. So you may consider that we have animal transportation, so we may have also opportunities for new cases in place or in countries that were previously non-endemic, but in which we have the vectors, and so the infection has spread very easily through Europe.
What's happened this year? Any big changes in who's getting infected and how because of the COVID situation? And has it changed how many animals are getting infected by parasites, or if they are?
You mean considering the transportation of animals or the limitation in transportation of animals?
Yeah, just in general this year. Have you seen any big changes?
In my work, my job, in my way of doing research, and also in the subject of in the research I do on that. Yeah, if it's correct, my understanding of your question, I would say that due to the limitations and the lockdown, limitation in the movements and lockdown, for sure, this kind of parasites do not have opportunity for spreading with animals to other countries. But due to the lockdown, we have less traffic around in the neighborhood. We have more wild animals coming into or around in the suburbs of the towns. So we could have more risk of infection with different parasites that we didn't experience, at least in in the urban areas. This is something that has to be considered, because of the increase in the population of foxes, for example, that are present in peri-urban areas that roam around. And so this is a scenario that could could have changed also during this period. As a researcher, I would say that our field trips are decreased, for sure, a lot. And this may be a bad thing. But by the other hand, we had more opportunities for working, for studying, for focusing on our research, the results of our research. So that was for me, a quite productive year in terms of publications and of researches carried out in the lab.
Of the parasites you're studying, which one to you is the most interesting and why? What about it?
So that's a difficult question. That's really a difficult question, because I would say that one of my favorite parasite was Thalassia callipeda eyeworm, because I studied and I discovered the biological life cycle and the vector, but also, for example, another parasite which is quite important, which is interesting, is also Onchocerca lupi, which is a parasite that in 2011 we found for the first time in Turkey to infect human beings. Onchocerca lupi usually is a parasite which lives inside the eye of dogs, but we demonstrated the zoonotic potential of this parasite. And after our first case in Turkey, many cases have been reported also in Greece, in North Africa, in United States as well. And our research and spreading research, and it's quite important for better knowing, but better appreciation of parasites that you may just misdiagnose, you may not know. Other parasites which are quite nice to be studied, and I studied quite a lot, are feline lungworms, which are some parasites which are snail-borne parasites living in the lungs of cats. And that's interesting because they represent a model for other snail-borne diseases, such as those of humans caused by Angiostrongylos costaricensis and cantonensis. In that case, we demonstrated a new path of infection of the third stage larvae, which are the infective larval stages, from a snail to another snail. And we named this new pattern of infection as intermediasis. Nowadays, we are we are working a lot on synanthropic reptiles like geckos and lizards as a reservoir of pathogens such as Borrelia and Rickettsia. We live in a world full of geckos, of lizards, but nobody works on those reptiles because they are difficult also to be catch and also to be sampled. And I find a lot of potentiality on that in this subject. And there was
is there any animal that you know of that doesn't have parasites? Any creature?
No.
Wow.
The parasites are everywhere, also in our society. We had a spot in Italy for people who didn't pay taxes and say, you are a parasite, a parasite of the society. Parasite is a concept. Parasite is everything eats or feed on another host at the expenses of another host. So it's a very, very old concept. And it was also a mask in the ancient Greek theater. And so parasite, every creature has a parasite.
When parasite goes from one host to another, I know in some of them there's asexual reproduction, in some there's sexual reproduction. What other main differences do you see when parasites switch hosts?
Okay. Sometimes some parasites are very host specific. Other parasites are less host specific, and the pressure of the environment, also of other selection factors, they made, in fact, more species of hosts. I can give you an example. Ixodes ricinus is the tick, the wood tick, which feeds on small rodents at larvae and the larval and nymphal stages, but also reptiles. Then it feeds on almost all animal species, including humans, and so they transmit in this way. They may transmit bacteria which are for which the reptiles or the rodents are typical reservoir. So host in which the parasite develop. And so you see, they may also change, parasites may also change in their evolutionary history, the host and host affiliation, but it depends on many factors and mainly, if you consider that it happens very slowly, we should live at least 4 or 5 times our lives for seeing a parasite which switch is a host, definitive host.
Well, very good, Domenico. What do you think you're going to be able to figure out in the next few years? Are you getting close to understanding any particular mechanism or parasite?
So we are studying the vector of these parasites of Onchocerca lupi, which is a parasite for which the biological cycle is not complete. For sure, we have different subjects to investigate. Scientists should be, in my opinion, more involved in the politics of stakeholders, decision making, and in the political decisions on some topical issues, such as, for example, the management of dogs or cats in a given environment. The role of also the veterinarians or veterinary parasitologists should change quite a lot, because we should be considered, and as we are, because I gave you an example. I teach at university in the Faculty of Medicine. We should be considered much more under the frame of One Health approach as a topical figure, professional figure for working for doing this kind of public health politics. What I am thinking what will be my favorite project, or what I will probably do in my next years. I don't know that. I would love to study and to learn and to know more on the role, for example, of reptiles as a reservoir of parasitic infections. And again, consider that nobody knows about these small creatures, but we have so many that in nature they must have some importance in the maintenance also of parasites. So this is something that is intriguing my mind, and I'm investing quite a lot on that.
You need to get a grant that sends you to like the Galapagos Islands for a year, and you could study the parasites of all these unusual creatures. You'd probably love it.
That would be great. Any researcher would be happy to work on that and to have a grant on that. And for example, that would be really nice to have supported projects in areas where usually nobody invests. For example, in Africa or in other developing countries. Just consider that 1% of research and development are invested in Africa compared to 40% in global research and development for veterinary parasitology funds in United States. So that's quite important. And I mean, I do not complain about funds, but it's a nightmare to have to work on that and to do applications continuously for having funds and for guarantee the young colleagues who work on with me at the university.
Very good, Domenico. Thank you for coming on the podcast and for your interest in what you do. I appreciate it.
Thank you to you, and we keep in touch. Okay, thank you for interviewing, and all the best.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.